Hello everyone, and welcome to our shark dissection. So today is the big day. So you should be working in groups of 2 to 3, depending on your preferences. You will have all of your resources here, but again, make sure that you've read through the shark dissection manual. This will be impossible to do unless you have read through all the pieces and know the organs and where you should expect to see them beforehand. And then make sure that you have a reliable group of people to work with where you've delineated tasks. Should be 1 to 2 people on the dissection itself. The shark is a large organism. And then 1 person taking notes, videoing the process, or taking pictures along the way. So with that, to your itemized checklist of things you need, I'll turn it over to Becca.
All right, so the very first thing on you guys' dissection for today is what materials you need. So I'll go over them real quick. We have our dissection tray, obviously, which is holding our sharks right now. A little rubber mat that our pins will go into. Here we have our T-pins, which we could use very liberally to help hold the shark down. As Paul said, this is a very large organism. You want to make sure that it stays where you want it to. We then have our dissecting needles, which you can use to pull around organs, point, whatever might be easiest for you. There's both bent, straight. Again, we've got options. Use what you're most comfortable with. We have scissors. Scissors are most often used for cutting open skin, cutting out organs, however that may work. We have the scalpel and razor blade. The razor blade is sharper than most of our scalpels, but if you're more comfortable using the scalpels, that's perfectly fine. These will be used for making first incisions, again, cutting out organs, moving skin. And we also have our tweezers. So there's both sharp tweezers and rounded ones. Use what you'd like. You can use them for pulling, moving, whatever you would really like to do with them.
Yep. And then we also have all the personal protective equipment, the PPE. I'd recommend an apron, as these sharks are fairly greasy. You'll obviously need a set of gloves if you're handling the dissection, and then of course some protective eyewear. So before we actually get to the shark dissection itself, the first thing we need to do is look at our 2 genders of our shark. So we've pulled out 2 sharks here, and they are of different genders. In order to gender the shark, you're going to be looking at its reproductive organs. So here we have a male with its claspers. These 2 structures, I'll let y'all read about them in the lab manual, but these are the claspers, and please be able to differentiate the claspers and the male cloaca from what you see on a female being these 2 fins and just the cloaca itself. Now, interestingly, this female has a prolapsed cloaca. It's fallen out of the female after death and infiltration with fluids. But you should be able to tell a very distinct difference between the claspers and the lack of claspers. Those are your sex organs. So with that now, I'm going to go ahead and return the male shark. We're going to come back and dissect the female. And we're going to be utilizing a dissecting microscope and all of our resources. So I'll see you in a few minutes to talk about the outside of the shark before we get to cutting its insides. All right, now we've got our single shark. So the first thing that I ask y'all to do before we get a little bit too messy is we're going to measure the shark. So Becca, can you go ahead and lay it out flat, or as flat as we can, just to get an idea of its overall length from its caudal fin to its head? What's the ultimate length we're looking at? About?
Roughly 68 centimeters.
All right, and so this is why your third person is necessary. So we're logging it as 68 centimeters. And of course, we've got our female here. Now, in terms of our structure on the outside, Becca, can you go over the fins that we're looking at?
Yes, of course. So, you guys are told to make sure you know all of the fins that we've got going on here. So we'll just go right down your list. We have the dorsal fins, anterior and posterior. So anterior means head, posterior means tail. So you have your anterior dorsal, your posterior dorsal here. Next,
dorsal meaning the back side of the spine, and what's the opposite of dorsal?
Ventral, which would be the bottom side.
Perfect.
Next, we'll look at our pectoral and pelvic. So pectoral, as would be on us, coming out of the chest, pectoral, and the pelvic, coming out of the pelvis, or down here, we have these. And lastly, we're looking at the caudal fin, which is another word for the tail fin.
Perfect. Now, as you're looking at these, you may notice that there are some really wicked looking spines. So these spines here are a defense organ for the shark, and they'll be coming out of both of the dorsal fins. Now, in terms of sensory organs, you may notice that the shark has a line down its body, and you notice the sandpapery feel. I do encourage you, if you want to, take your glove off and feel the shark. I've done this many times. It feels and sounds like sandpaper. I don't know if y'all can hear this. Very textured skin, but you have the lateral line here. This is a feature made up of tiny pores that have the ability for the shark to sense sensation at a distance. So it's touch at a distance. When we're done with this outer analysis, we're going to actually take a section of the lateral line and throw it under the dissecting microscope, so that way y'all can see what it looks like there. Now, from there, let's talk a little bit about the structures in the head.
So up in the head, first thing we're going to look at is the spiracles, which are right here above the eyes. There's an opening there and an opening here on each side.
The two spiracles on the top side of the head, and then we'll flip the shark over here to look at the nostrils of the shark. So obviously, if you've watched movies, we know that sharks do have a sense of smell, even though sense of smell in water is very different from sense of smell in air. The nostrils here allow for those sensory receptors to contact the stuff in the water. Now on top of that, or actually just beneath that, you have the ampullae of Lorenzini. These sense organs are similar to the lateral line, and we're also going to be cutting out a piece of them to throw them under the dissecting microscopes to look at the channels found there. But this is basically where a shark is detecting where to bite. This helps it feel out what's directly ahead of it, because as you'll notice, its eyeballs are kind of on the sides of its head, and its biting needs to happen here. So there is kind of a disconnect where the shark is biting by feeling, but still being able to see things, even if only moderately well, at a distance with its eyeballs. And then the last thing on the outside
is we're going to be looking at the cloaca of the shark, which is both, comes into a couple of your different systems, as you guys will have read about, which is going to be right down in here.
And this would be a hole that would be pressed back inside. We see some of it extruding, but it's down by these pelvic fins. So with that, Becca and I will actually get to cutting a small section out of the ampullae of Lorenzini, as well as out of the lateral line, and we have our dissecting microscope here. Once that's done, we'll see you guys at the scope to take a look what they look like underneath the dissecting microscope. See you.
So here we have the placoid scales on the outside of the shark, and then a close-up on the ampullae of Lorenzini. You can see the lighter spots. Now, it's important to take a look at these structures, the skin of the shark, to see the homology that they have and overall texture with some of our other structures in later vertebrates. Now, these cells actually have tiny little blood vessels running into each of them, and they're covered in material similar to that of our teeth. Because yes, sharks' placoid scales share ancestry with vertebrate teeth. Just like the scales, each of our teeth have a blood vessel running into it, and they grow or develop in similar patterns. Now, there has been a lot of change, like our skin is not our teeth anymore, but it is cool to note this phenology in terms of phenology and ancestry in terms of how they look and what they come from. Now, another interesting thing about these shark scales is that they will only produce more of them rather than getting bigger in size as the shark gets bigger. So it's a numbers game. As the shark gets bigger, you just produce more of these placoid scales to help cover up the skin and reduce the drag and all the other components that these scales help in swimming. Such as, actually, the model for these placoid scales was used in trying to prevent the hulls of ships from over-encrusting with barnacles and things. So there are other potential microbial benefits to these placoid scales.
All right, so now I can show y'all where we've cut the incision for the lateral line that y'all saw under the dissecting microscopes, as well as the ampullae of Lorenzini. And at the same time that we're doing that, we cut open the side of the shark, so that way you can see some of the individual structures of the myomeres, the stacked muscles that allow the shark to swim left to right in terms of its overall movement. And then at the same time, just out of convenience sakes, because the spines on this shark were kind of long, we cut through the spines. It's very difficult to cut through these on account of the fact that they are cartilaginous, but the other option is just be very safe when you're moving the shark around. So next up, we are going to flip the shark over. And then the instructions say to make a direct incision from the cloaca, opening up the digestive cavity to approximately the bottom of the jaw. So I'd recommend you go not quite, you can go all the way up right now, but you'll also notice that you see the shark's gills here. We'll be dissecting the gills very carefully after we get through the digestive cavity. So for now, you might want to stop more towards the pectoral side of the shark. Now your shark may also have some staples as they, stained the insides of the shark to help keep things identifiable in terms of color. You can feel free to remove them here, but know that your razor blade will not be able to cut through those staples. So you can pin the shark down or just go for, have one individual holding it. I will be holding on one end, Becca will hold on the other, and we will start with the incision from the cloaca all the way down to where the incisions were made for coloring. Now again, I prefer to use the razor blade as I find it to be the sharpest of the tools, but you may want to utilize other cutting devices. Might take you a bit to get through the muscle, or it might go quickly, just like that. Now it's about being able to separate the staples. This here might need a little bit of tearing. I'd recommend not ripping the flesh open. I'd rather just move things through here, cut around the edges of the staples. This is where your scissors can come very much in handy. Thank you for that, Becca. So now, I'll also cut diagonal here, again through the muscular tissue, to kind of create some open flaps. Be very conscious of your classmates' appendages. This can be quite dangerous. Last thing I want to do is see any human blood involved in this. But here we have the open cavity of our spiny dogfish shark. So now we can go ahead and pin this down. So Becca, would you mind grabbing some pins? And this is the purpose of the rubber mat, to help keep the shark pinned down. You'll notice that the rigor mortis has definitely set in. It is stuck in its way, keeping in mind that this shark came from a bucket where it was stored in, so it's kind of stuck in that bent shape. But hopefully these pins can at least keep the flaps down, so that way we can identify all of our different organs. If you need to, you can go ahead and widen any of the cuts as necessary and use as many pins as necessary. So here now, what we're going to do before we discuss any of the organs, I want y'all to take a look at the internal structures of the shark. I won't tell you what anything is at first. I'm just going to move stuff around, and then we're going to go ahead and pause the video and get back and identify all of these different structures. So just moving our different structures around, so that way you can identify these different components. This piece is all connected. This piece is ancillary, quite hard, several lobes. Structures back there. So yeah, we will pause the video here, give you guys a chance to identify or see if you can identify any of these different organs. All right, so now hopefully you've had a chance to look at these organs. Now we're going to go over them together. So this is obviously the largest organ, several lobed, two big ones, one small one. Becca, what is this large organ?
This is the liver.
What a giant liver. Now in the liver, you'll have attached the gallbladder. Now unfortunately, I've done a little bit of a number on the gallbladder and trying to cut it up, but you can see in trying to cut the shark open, but you can see where the gallbladder was as it's connected to our bile duct, stained in yellow here, leading into the digestive system. The digestive system itself, you'll probably get this one better by feeling into it, but you have the esophagus up here, and then we have the stomach proper. Now, after we leave the stomach, we have the pyloric valve and the duodenum with its lighter pink rings. Things will be moved along in the digestive tract into the ileum, where you start seeing the more pink rings with some yellow surrounding it, and then down into the colon and the cloaca, which again on this individual has prolapsed. Now, ancillary organs to this, we have this gray knobby thing here. That is our spleen. And then our next task, oh, and the pancreas, the filmy structure here at the bottom. This is where you're going to be secreting a lot of hormones into the bloodstream, so that way the shark can regulate while it's digesting. And then next up, we have unique female features. So we're going to be looking at these in a second, but you have these very hard, round structures. And then we need to next go inside the stomach. So Becca, we've found ourselves a specimen in the stomach, so we'd like to pull that out live on camera here. It looks like this shark was eating another fish. Fish fragments, different scales, part of the spine, kind of gross. Now inside the stomach, because the shark does not chew with its mouth, it throws it straight down, you will see the mucosa and rugae. These structures help provide extra surface area and moving around for the thing as it's being digested, because as you just saw, food is thrown in whole into the shark's stomach, and those mucosa and rugae are going to be helping it digest. You'll notice that they get much, much more structured towards the exit, the pyloric valve of the shark's stomach. This is the valve that's going to prevent the food from going down into the digestive tract before it's ready to do so. Now, next up, we're going to be moving away from our digestive system to take a look at some of the reproductive structures. So Becca, would you be so kind as to remove one of these large orbs?
Happily.
Oh, just pulled it directly out. Go ahead and cut that in half to take a look at what's inside of there. As these are the sacs from which the young would develop, they are just giant balls of what would one day evolve into yolk, large fat sacs. Now this female, how many of these eggs did she have ready to go for fertilization?
We found 4 of them this size up in the chest cavity.
Crazy. So that, the size of these, comparable, is going to be as large or larger than the digestive tract. So that is such a huge sacrifice, huge commitment to reproduction that the female has to have. Now, if you want to, you can also trace the ducts that would release these sharks, because these sharks would incubate inside of her for a while in a way that's very different from our placenta. These sharks live off of the fat in the sacs rather than continuing nutrition from the mother, and then will be released through the reproductive tracts here and out, so that way they can go on swimming. Now, next up, we're gonna go through and cut open the shark's breathing system, its gill slits. So we'll remove the organs, move aside some things to kind of clean up the space, and then we'll see you guys back here in a few minutes for our dissection of its gills. Okay, so now, after we've removed all the internal organs, we spent a good 20-ish minutes making our next incisions. Becca doing most of the legwork here. It can be really difficult to dissect the parts around the jaw. You're going to be cutting through some cartilage, so please be very, very safe with all of this. But let's take a look at the product of all this. So you'll notice, first and foremost, that we dissected just beneath the jaw and cut down to the gill slits. We haven't connected all the way through, but you can. You'll notice here you have the two jaw arches, the mandibular arch and the hyroid arch, and the triangular teeth that are around this. Now, actually, in a funny story, when dissecting the jaw here, Becca's glove got caught on some of those teeth and actually stayed in the shark's mouth. So you want to be careful with these. They are kind of Velcro-y in the fact that they will be able to rip your gloves off. So be careful. You see the tongue of the shark, the mouth muscle down there. You also see the openings to the spiracles. So in the back here, see if you can see that? Yeah, there we go. Two spiracle openings there and there. And then you have the structures around the gills. Now the gills are fascinating because these are the early breathing structures. So you're going to have the openings to the gills itself where water will be coming through. Now remember, the whole purpose of these is to get oxygen. So you're going to need a lot of surface area. So when we look inside the gills, you have three major structures. The gill rakers that give this appearance of like the endless teeth inside the shark's mouth. So that way they are actually catching any of the small prey that may have gotten into the gills. This is obviously not such a big problem for a small shark like this, but for a bigger organism, the gill rakers will be more pronounced. And then we have the arches, each of these individual lines that are providing kind of the backbone for the gills themselves. And then the filaments, each individual flap, here stained pink, where the gas exchange is actually happening. So you have hundreds of little filaments arrayed one next to the other to get the shark enough oxygen. Oh, you okay? Mm-hmm. That pin just popped out. So those filaments are going to be spread around in order to get the shark enough oxygen. And then they're supported on the arches with the rakers providing protection against small animals getting stuck in your lungs, or the lung equivalent, because these are what fish and sharks use in order to breathe. Now, once the oxygenated blood has been absorbed into, or the oxygen has been absorbed into the shark's bloodstream through the gills, we then are going to take a look at our heart system. So we're going to open up the chest cavity here and see y'all back in just a second to take a look at our shark heart. All right, so here we are with a couple hands holding open the shark's rib cage. Now the shark skeleton is made of cartilage, so you should be able to cut through that, though it might be a little bit of work. And then we've got probably the most important organ on the shark, its heart. So you notice that there is a difference in the staining on the heart, as well as if you're touching it, you can see or feel a difference in the texture. You probably can even see it. You have one bag that is much more loose, and then one bag that is much harder. And these corresponds to the two different chambers of the heart. The atrium, that is collecting the blood from the body, and the ventricle, that is then taking the blood and pumping it towards the gills, where it's going to be getting oxygenated, and then carries it over to the rest of the body. So the ventricle is actually the thick-walled structure here. It's going to pump blood through the conus arterius. This here, a cone-shaped artery that's going to lead to the gills and then the body. Inside of this, see if we can observe. So you do see some of the blood vessels. The blood vessels that are now separating, heading off to each of the gills where they're going to be oxygenated. But if you cut this open, you may even be able to see some of these semilunar valves. Cut this open, and the semilunar valves along the way, I don't know if they're going to come out in this shot, that's fine, but they're basically helping. So you can see how there's a divot here. Looks like a little half moon. See if we can get that to come through. each of those are going to start spreading the blood to eat the different sides of the gills, where it's going to be gathering its oxygen. So there we have one valve, and you can continue up the way there. And then you have the efferent and afferent blood vessels that are going to be sending blood into the heart, into the lungs, or the gills rather, in order to be oxygenated. And so this is the general body plan. A single circuit loop where the heart is going to pool all of the blood from the body, and with a weak muscle that has to only pump it ever so far to the ventricle, where the ventricle is a much stronger muscle that then pumps the blood into the gills in order to be oxygenated. And so there we have our shark dissection. We hopefully have treated the shark with respect and dissected and removed all of the internal organs. So as a last review, here's what the cavity looks like when it's empty. We do have 2 undeveloped eggs still within the oviducts. Here we have all of the, on the side here, you'll see the 5 eggs. We thought that there were only 4. We found another one on the underside of the shark. So the eggs there. We have the large lobes of the liver with the bile duct still intact, the gallbladder attached at the top there. And then we have the digestive system of the shark, starting with the stomach with its mucosa and rugae to help with digestion, leading into the duodenum with the pancreas attached as well, and then the ileum, where a lot of the digestion will happen. So from here, please make sure to wash out your tray. All of your trash is going to need to go into one trash bin committed for all of the shark wastes here. We want to be respectful of maintenance in terms of cleaning up the place. Once your shark is disposed of in the designated trash can, please make sure to wash off all of your utensils for the reusable stuff, like our scalpels and our tweezers, etc. Please wash them first in soap, and then you're going to leave them in the middle, the middle table with the faucets for a ethanol wash afterwards. Same goes for your tray. So please wash them off with soap and water first, then leave them in the middle hexagonal table for ethanol wash after that. All gloves, all shark wastes into that one designated trash can, and then share your photo documentation of the lab, and have a good one.
So now that we're done with our shark dissection, I'd like to revisit these models, because it's important to understand where all of these organs are in relation to one another, as well as how they compare to other organisms. The final part of this lab is homework, is going through and defining the function of all of the different organs that were highlighted in bold letters in the lab. As you go through this, put little asterisks in things that have retained similar function. Note where there are large homologous structures and ancestries that still perform similar uses in vastly diverse organisms, whether they be in sharks or whether they be in frogs. Now, just as important as the similarities, please note some of the lacking organs in a shark, or some of the organs that do not persist into other lineages, like lungs being an entirely new thing. Though you may have noticed in the dissection that you see something similar to the diaphragm, a structure at the base of the rib cage of the shark, protecting the heart, that would have separated everything within the rib cage from everything beneath the rib cage. So even in new structures, they sometimes can be developments on materials that were already present. In fact, they always are. So see where you can trace these different ancestries within our sharks, our frogs, and our rats, as well as yourself.